I'm recording this now, but this is not on the record. So don't hold me to this at the end of the semester. But just looking at this, I'm thinking that this is something like A's, B's, C's, and then somewhere down in like the 210 range might be, you should be concerned about whether or not you're going to pass at this point. And remember, your lowest grade on an exam will get thrown out. So if I was going to curve this class right now, this is kind of letter grade distribution. Okay. Now, so you can discuss that with each other later if you want. I'm sure you will. There were a couple of questions, which I'm not going to show you these data right now, but at the end of every exam, I look at every question, and one of the most important questions to me when I look at your scores as a whole is, was there any question I wrote where not a single person got all the points? And there was one of those questions on this exam, which means at the very least I did a very bad job of teaching you that point, or I wrote a very tricky, misleading question that was not fair. So in a subsequent class, or maybe as a post on Blackboard, we'll see, I'm going to tell you which questions are the ones where it looks like I failed. Those questions are questions that are going to show up again at some point, probably on the final. Right? Specifically those, not necessarily those questions as written, but the topics of those questions. I'm going to retest at least on the final. So it'll be useful if I point out which questions those are for you to make sure that you've reviewed the exam key and understand what I was looking for, so that when I ask a similar type of question again at the end of the semester, you're prepared to know what I'm trying to test on. That's the tricky part for you, I think, as students. It's obvious to me when I write a question, the key concept I'm trying to ask you about, and it's difficult for you to figure out, while it's a stressful environment, 50 minutes, wait, what is the important point that I'm supposed to be writing about or answering here? So that's what I'm trying to get you to think about when I tell you which questions these are, is to go in and look at those questions and try to figure out, you know, what was the key concept, what was the thing that I could have answered differently. Okay, let's take a look at some quiz answers. So, dihybrid test cross. Looks like the common answer is A. What's the, let's see. But the correct answer is B. Okay. So maybe we should look at the next question. Hold on, just a second. Let's see if we get the same pattern of answers. Yes. So what's the difference? So everybody said A on the first, most people said A on the first and A on the second. This one is correct. This is the dihybrid cross. So definition of a dihybrid? Anybody? Heterozygous at two loci. Di, two loci, hybrid, heterozygous. So that's what that is. That's two different heterozygotes, F1 over F2, G1 over G2, crossed to itself. So how does that differ from this question? What's different about the second genotype? This is a test cross. We have a dihybrid, and then we need a tester individual. Someone who's homozygous at every locus. Some individual that's homozygous at every locus. So that's why this answer is B. The first individual's dihybrid, the second one is homozygous at both loci. What is a common use of a dihybrid cross? Actually, all of these are possible uses of dihybrid crosses. Which is totally fine. It doesn't matter if you answered 1, 2, 3, or 4. All 4, that is. But these are all things that you can do with the dihybrid cross. And that's what we're here today to talk about, in part, is the outcome of setting up a dihybrid cross, looking at the genotypes and phenotypes that result from it. was the last question? Oh, yes. Good point. How many gamete haplotypes are produced? Ooh, complicated. 3 loci. We haven't talked about 3 loci before. But it could be anything. Could be any of these options. No, that's not true. It could be. There's one answer. This is one where there is one answer. So I added this distractor. You've never seen, well, there are lots of distractor choices. I'm talking about in the genotype. What's misleading about this super complicated 3-locus genotype that I've thrown at you?
The 3 locus doesn't necessarily
One of the 3 loci is homozygous, right? B2 over B2. So no matter what, that individual is going to contribute a B2 allele to their offspring. We go old school. Let's go old school. What's the C? C1 over C3. So how many different ways can you make combinations of all of those letters? 4. If you have A1, you're definitely going to have a B2. You could either have C1 or you could have C3. Or you have A2 up front. You're still going to have B2 at that locus. And then again, you either have C1 or C3. It was basically a dihybrid cross because this is constant regardless of what combination of A's and C's you have. So I guess that makes the answer 4. Now you know. So, wait a second. Which got the most votes? Wait a second. Is that changing?
Yes.
That's okay. It was D, right?
Yeah.
Initially, I should have taken a screenshot. What's the answer if this is a trihybrid? Then it's 8. Because you were so close. Yeah. If you have 2 choices, this is just like probabilities. If you have 2 choices there and 2 choices of genotype there and 2 choices of genotype here, you've got 2 times 2 times 2 or 8 different combinations. It's like the lottery? This is better than the lottery. No. Okay, so that's, wow, look at those answers change. There you go. All right. Now I think maybe we're ready for this. So for videos to watch today, related. This is more complicated than it seems at first blush. Draw a Punnett square. 2 individuals looking at 2 loci each. So how do we set this up? How many different gametes will that individual produce? A dihybrid. How many gametes? So there's an individual, A1 over A2, B1 over B2. It's a dihybrid genotype. So its gametes will have A1 with B1. What else? A1 with B2, A2 with B1, and A2 with B2. So there's the start of a Punnett square. Is this going to be a 4 by 4 Punnett square? So how many rows? How many different haplotypes does that red individual produce? Is it A1 or A1? Every gamete's going to have one version of gene A, haploid, and one version of gene B. So the version of gene A they get has to be 1. And gene B, we're looking over here. There's, thank you. Voices from the middle of the class and beyond. So A1 and B2. 100% of the gametes. The only choice from that parent, it's not a choice. They only have A1 genes. They only have B2 versions of that gene. So they can only make gametes that have A1s and B2s, 100% of the gametes. So that's it, one row in the Punnett square. You could draw it 4 times if you want and make a 4x4 Punnett square, but it gives you the exact same information.
So you were saying that all of those are in one individual, or are we crossing two separate individuals?
So this is individual 1 in black and the individual 2 in red.
So 1 is hetero while 2 is homo?
That's correct. The individual 1 here is what kind of a genotype? What one word do we use for that genotype? The individual in black. This is a dihybrid, heterozygous at 2 loci. And that's why we have 4 columns, because a dihybrid produces 4 different types of gametes, 4 genetically distinct haplotypes. But somebody who's homozygous at every locus we're looking at, like individual 2, produces genetically homogeneous gametes, only one type. So you could write that 4 times down the left side and fill in a 16 square Punnett square, save yourself 75% of the work, and just write the one type of gamete they produce. So then you could fill that in if you want.
Yes, please, can you fill it in?
Okay. So that's A1 over A1 and B1 over B1. B1 over B2, sorry. So are these going to be all different genotypes? That's the question I want to know. Good grief. Hang on. How many different genotypes do we get here? 4. So far we've got the 2 individuals in the cells are both homozygous A1, but they differ at the B gene. Okay, now we're going to see heterozygosity at A and B, and then finally heterozygosity at the A gene and homozygosity for allele 2 at B. So are any 2 of those genotypes the same, or are all 4 of those different? We've got 4 different genotypes. So they're present in a 1 to 1 to 1 to 1 ratio. For every 1 of individual W, there's 1 with genotype X, 1 with genotype Y, and 1 with genotype Z, for those of you that are British or Canadian. And that's only because there are 4 different gametes produced by the dihybrid, and they exist in a 1 to 1 to 1 to 1 ratio. And at some point we will fill out, maybe, the full 16 square Punnett square and look at all of its glory, but we don't need to do that right now. Bless you.
Is there a reason you need to do a whole Punnett square instead of just doing that?
All right, I think one more question from the pre-lecture video. Be a forensic geneticist. Try to guess. This is leading us into where we're heading in a couple of class periods. We're going to start looking at pedigree analysis, which I think is one of the most fun things, despite what you think right now. And one of the most interesting and fun parts about genetics. If you like solving logic problems, which I'm not saying you have to, if you like solving logic problems, pedigree analysis is where it's at. And this is part of that. So if you've got a genotype that's C2 over C7, that's one gene, and D1 over D1, what can we guess about the parents? We've got a couple of parent genotypes. So they both, well, they each have one, they each have to have a D1 because each parent has to have an allele 1 of gene D to contribute to a kid if the kid's genotype is going to be D1 over D1. Okay. Do we know anything about what's on the left side of the slash? Not yet. We just know that each parent has one version of D1. Likewise, you know that at the C locus, one parent has C2 and one parent has the 7th allele of gene C. But again, we don't know what their other allele is. What other information could be useful to figure this out that we could collect if this was a study we were doing? Humans, dogs, chimpanzees, rats, orangutans, any species. What would we need to know to figure out if we didn't have the parents, if we had access to siblings? So what do we need? We need more sibling genotypes. So that's one individual. Let's say this is an individual, this happened to me a couple of years ago. Somebody walks into my office and says, this was a really strange conversation. Walks in from off the street, literally. Had been wandering around the science building, biology, chemistry building, looking for somebody with genetics expertise. Has a lady friend, long story. I can't make it shorter either. Basically, he was trying to prove that his lady friend, this is an older gentleman, wanted to prove that his lady friend is the daughter of an American citizen. She's abroad right now. Apparently she'd been kidnapped early in her life and taken overseas under a false birth certificate or passport. And so she was currently living in Britain, and the State Department wouldn't give her a US passport. So this guy was trying to prove that she was the daughter of a US citizen so that she would be eligible to get a US passport so that she could come back to the US. Can't make this stuff up, or maybe you can, I don't know. But it seemed legit at the time. And what he needed then was genotypes. He could get her DNA. She would submit her DNA to get genotyped. He would need the genotypes of siblings. If her parents are, one parent was dead, I think, one was still living. So you would need additional genetic information to prove that she was a member of that family. You might want to know, what are the siblings? So I'm going to draw a couple of things in right now, and then we'll leave it at that for today. You can try this as a practice exercise if you want to work on figuring this out. But let's say her siblings' genotypes are, so just work on this later if you want. C2 over C2 and D1 over D2. And let's say over here we've got D2 over D2 and C2 over C7. Okay, so we'll leave it at that. Say you get that information. Now can we unambiguously, without a doubt, figure out what the genotypes of the parents are? So we'll talk about that later. So it may be, okay, everybody okay with this information? It's recorded, but it's also here now if you want to write it down. That was one of the other questions that we're catching up on questions that I asked last class, what you wanted to know about. So spent a lot of last class talking about other questions. Now we're finally getting to last class's content. That's basically this question. So I asked, is this something you'd like to know? A lot of you said yes. So in what situations do you see Punnett squares that aren't 2 by 2 or 4 by 4? So that's a 4 square, a 16 square Punnett square. So what's the situation where you have a 4 cell? What sort of a cross is that normally? It's a monohybrid. So if you have something like a heterozygote crossed to a heterozygote, then you've got two different types of gametes, two different versions of gene A, and that's why this square is divided in half vertically and in half horizontally, because each parent has two different gametes they produce. The exact same is true for the 16 square Punnett square. That's a dihybrid cross, like we just saw. So that's 4 haplotypes per parent. And that's why each axis has 4 columns, 4 rows. So what happens if we look at a trihybrid? Extrapolate this information. If I ever asked you to draw a trihybrid Punnett square, right, this goes in, this is 4 is 2 squared. That's 2 alleles at each locus. We're talking about 2 loci. So that's 2 alleles, 2 loci. 16 square is 2 cubed. That's still 2 alleles per locus, but now we're talking about 3 loci. Sorry, 2 squared is 4. That's, yeah, 2 parent, 2 loci. You're right. The math doesn't work out. But the answer was correct. The next one is a 64 square Punnett square, which has 8 columns and 8 rows, which I will never, ever, ever, ever ask you to do anything with in class, just to make the point that the more loci you look at, the more complicated things get. That's all you need to take from this whole slide. That's why normally we stop talking about dihybrid crosses. You can do the rest. You could do a tetrahybrid cross and look at 256 square Punnett squares, but what the hell's the point? So don't worry about the arithmetic, just worry about this pattern. So you get a 2 by 2 when you're looking at one locus, you get a 4 by 4 when you're looking at dihybrids at 2 loci. What are the exceptions then? We just saw one. If any of these loci become homozygous, then you don't have to have a square anymore, you can have a rectangle. So we just looked at one that looked like that. This was a dihybrid parent, and there was a homozygote. Scroll back through the movie if you want to find it. There was a homozygote and a dihybrid. So we only had one type of allele from one parent, one type of haplotype from one parent. You don't need all those extra rows, you just need the columns for one parent's haplotypes. I think we already looked at another type. If you look at a monohybrid cross, right, that's a 2-cell Punnett square. That was today. A1 and A2 from that parent, separate haplotypes. And this parent over here only makes one type. So you don't need a 4 by 4, you can use just one row. Bless. Yeah, I know, it's that time of year. The plants are having sex. Or trying to, anyway. Meiosis and all this, sorry, genetics jokes. Maybe somebody could come up with one for Comedy Genius. Yes?
So just for clarification,
Please.
what circumstances would you see Punnett squares not divided to 4 or 16? The answer would be when you have homozygous link.
When you have homozygous genotypes, then that'll reduce the number of gametes, different gametes that are produced.
So, assuming you are doing a dihybrid cross in which both parents are heterozygous,
Okay.
I was filling one out a couple nights ago, and I noticed like trends in which like you kind of get these diagonal lines in which
So the genotypes in the middle of these 16 square Punnett squares, if you do a dihybrid cross, so let's say you selfed a dihybrid and you produce that 16 square Punnett square. Whether or not you see patterns in the genotypes when you fill this in depends on the order in which you write the haplotypes down and across the axis. So if you do those if you write them in order left to right and top to bottom in a systematic way, you'll see patterns. Otherwise, if you write them sort of haphazardly, that won't be as obvious. But there is a mathematical pattern in this process, and we're going to look at that in a future, but very near future class. We're going to start talking about the mathematics of that Punnett square profile. Oh man, did I distract you all now? I'm sorry. Okay. I had one student, thank you very much, I appreciate it. More would be super excited to receive. I had one student email a question. Remember, at the start of last class, I said what I'd love to be able to do is have you email questions in, we could vote on them at the start of class, which ones I'll answer during class. Maybe a good idea, maybe not, but I got one submission today, which is a great question, I think. So here we go. We've already talked about some principles related to this, but dihybrid cross. We're about to look at the C. elegans roller GFP question that we've been talking about for a couple of classes now. I want to come to an answer on that one for you today. The traits are on separate chromosomes. We've got GFP on one chromosome, we've got the roller gene on another chromosome. We're trying to make those GFP roller wavy worms. All of the examples so far in class have been genes on separate chromosomes. Hence this question. Can we ever, as geneticists, combine traits that are controlled by genes that are on the same chromosome?
Couldn't you say that possibly from the example we did were from traits on the same chromosome, we just labeled them as A and B?
Right. So if I was going to draw out and pose this problem to you, this is the way I would do it. And again, starting next week, I'm going to try to avoid a little bit of detail now so that we can save some for next week. This has everything to do with recombination and what that does to genes that are linked. So two genes that are on the same chromosome. Let's say, for example, the roller gene is there, and you have two minus alleles. Those are the mutant versions. And let's say you've got GFP on one copy of the chromosome, but not the other. So this is basically the question. Those genes are now linked. Or we have the situation that we're considering right now where they're unlinked. They're on two separate chromosomes. Does it matter if they're on the same chromosome or not?
Can't you have like dependent and then independent ones kind of?
The answer, does it have to be different? That's the really important question for us just to come to a consensus on right now. For us to combine traits, do they have to be on different chromosomes?
No.
No. So they can be on the same chromosome, and we'll see that as we enter the next couple of classes.
But I mean, if
Go
they're
ahead.
linked, then where one goes, the other one will go. So essentially, it's still a monohybrid cross.
So this is the interesting thing, yeah. So from the question posed yet another way is, could you ever make, if you just had one worm, it's a hermaphrodite, let's say. So you have that genotype. If those genes are on the same chromosome, could you ever make a worm that is not green, but a roller? So what would the genotype, think for a second, what would the genotype be of a non-green roller? So we said the roller was recessive, so you have to have minus minus to be a roller. And what's the genotype to be not green? You have to have no asterisks, like no copies of the GFP gene. So you'd have to be plus over plus. That's non-green, or wild type, if you will. So that would be the genotype of a non-green worm that spins around in a spiral, a corkscrew, when it tries to crawl. Can we ever get that with that genotype circled on the left?
If they're far enough apart, can't that happen with crossing over during
And that's where we're headed with the next topic on the syllabus is when crossing over happens between the genes. So the closer the genes are, in brief answer to the question, then we move on. Just wanted to make sure that you've all heard this once because this is, again, like pedigree analysis, where we're heading next in class. The closer together two genes are, the more likely they are they go together at meiosis into the same gamete. And if the two genes that control the two traits you're trying to combine are next to each other on the chromosome, when they're really tightly linked, then they always travel together as a package. So in this case, you might argue that if those two genes are literally right next to each other on the chromosome, the GFP gene and the gene that makes a worm roll, that you can't separate those. That you're always, if you have a green worm, it's going to have the roller mutation. If it's got the roller mutation, you're going to have the GFP gene. So the common assumption is, right, it's easiest, this is absolutely something you should write. It is certainly easiest to combine traits when they're on separate chromosomes. Because when they're on separate chromosomes, that's when independent assortment happens, and you get nice ratios like 1 to 1 to 1 to 1 haplotypes out of a dihybrid. Linkage screws all sorts of things up and makes life difficult.
I have a question.
Please.
Are we just not writing the pluses or for the non-green? Or do they just count as those little, up there? Yeah, okay.
So here's a plus. There you go. So normally I omit wild type nomenclature. That's the default. Remember, anytime you see a question, if I haven't specified a genotype, you should assume it's wild type or plus. Oh, and look, now the plus looks like a chromosome with a gene on it. That's annoying. Make those chromosomes longer. Okay.
So the answer would be that they could be on separate or the same chromosome.
Yes. It's possible to combine two traits that are on separate chromosomes or the same chromosome. The short answer is it's easier if traits you're interested in making new combinations of are on different chromosomes.
But if the two genes are too close, crossing over would be,
Right.
like there has to be.
So we'll get to crossing over in the near future. And I'll try to set up, there are two questions left that I've posed in previous classes I want to make sure I get to today before we leave these topics behind, so that you have the answers in case you've been wondering when we're going to get to them. But the next question we're going to look at, we can answer in two different ways. We can ask whether the genes are linked or unlinked. This one, not so much. This is a more straightforward question. So there's a really critical point to take away from this question. We're going to talk about genotype ratios and phenotype ratios, the difference between the two, which are really critical. And this is one thing to pay attention to on future exams. When I ask for a ratio, make sure you look at the word before ratio, which will either say genotype or phenotype, and make sure that you're addressing the correct type of ratio. So this asks for genotypes and phenotypes. So what does this Punnett square look like? So first, let's answer the last question. How many genes are we looking at? We're just looking at GFP. Yeah, forgetting the roller for right now. So just GFP. We've got this genotype. Asterisk over plus. So if we self that individual, that's the equivalent of a monohybrid cross. It's crossing an individual with that genotype by itself, which by definition itself has the same genotype. So what does the Punnett square look like? What are the dimensions? Each of those parents can create, well, it's the same parent. You either get an asterisk or a plus. It's a simple monohybrid Punnett square. So we're going to get that ratio of offspring. So we've got 4 different cells. How many different genotypes did we produce in the next generation? The genotypes that are inside the Punnett square. So what are the 3? I heard 3. What are the 3? There are 2 homozygous. Those are there and there. Those are the heterozygotes. So there's 2 of those. And then what? So one homozygous in red here. There's 1 of those. And then the only other one left is wild type. And that's where we get that common ratio 1 to 2 to 1. 1 homozygous, 1 allele, 2 heterozygotes, and 1 homozygous, the other allele. That's genotype ratio. Not shockingly, because we're looking at the genotypes. This is the first time we've been able to look in class now at the phenotypes. Right? Genotype is a description of what the genes are. Phenotype is the description of what the organism looks like. So how many green fluorescent worms are there? Which ones are those? Anybody that's got an asterisk, you're right. 1 or 2, it doesn't matter, will be GFP. Sorry, I should have chosen green, I suppose, if I actually knew what green looked like. So I stick with yellow instead. So we have 3 green fluorescent worms. Don't make fun of me, I've got a serious medical disease. It's a genetic disorder, it's called color blindness. And then we've got 1 worm that's wild type. That's the 3 to 1 ratio. So dihybrid cross, don't have to memorize this, you should be able to recreate it for yourselves, but the genotype ratio from the monohybrid cross is 1 to 2 to 1. And the reason there's a 3 to 1 ratio is because you add those 2 genotypes, 1 part homozygous GFP and 2 parts heterozygous, to tell you the total number of worms that have GFP, green. So 1 to 2 to 1 genotype ratio, 3 to 1 phenotype ratio, those are important to be able to distinguish. So now we figured out the answer to the first question, which is what are the offspring that get produced by this cross?
So if that's the genotype ratio, the phenotype ratio would be 3 to 1?
3 to 1. So I should write that 1 over here somewhere. 1 wild type. 3 GFP to 1 wild type. So this was the big question. Your job, classes ago, when we actually posed this question. So you've got roller worms, they're minus over minus. We assume that their genotype, because I didn't describe it, was plus over plus for GFP, while not green. And you have some GFP worms, they're green because they're heterozygous, but they're not rollers. So those are your 2 genotypes. So let's say we cross these worms together. Use all the information you've already practiced today to set up the Punnett square. What happens when we cross these worms together? We call this top worm worm 1, and the second worm, the second genotype, worm 2. They're in upper left in blue. So what happens when you have worm 1 create gametes? What are its haplotypes? We're going to set up a Punnett square over here. How many different haplotypes? What sort of gametes does that worm produce? Minus at the roll locus, plus for GFP. So how many columns in our Punnett square?
4.
Well, 4 normally, but we've only got one type of genotype. So it's going to be a very tall and skinny Punnett square. Maybe. Now what about worm 2? Let's put its gamete haplotypes over here. So it's always going to have a plus at the roller locus. And it either has an asterisk at GFP or it's got a plus at GFP. So complicated looking cross turns out to be a 1×2 Punnett square. We have 2 different types of genotypes of their offspring. Which one's more useful for getting to our goal? The goal really is the answer to question 3. So let's skip down there for a second. Sorry. Question 2. This thing I'm underlining. What would the genotype be of the worm we're trying to get to? It's always useful to know where you're heading. So you want a worm with both traits, a roller that's green, that only produces all of its offspring are also green and rolly. So what's the ideal genotype to do that? Homozygous for both, right? Pure breeding. It only produces offspring that look like itself. So what is that genotype in our nomenclature? Asterisk over asterisk, minus over minus. So that's where we're trying to get to. We're trying to get a worm that only has asterisks and only has minuses. Up here, if we just cross these 2 worms together we were given, do we reach that goal? No. Which one comes closer, the genotype on the top or the genotype on the bottom? Do I need to open Socrative again? Why is it top? It's heterozygous for both. So this worm in red at least has 1 minus and 1 asterisk. It's kind of like we're halfway there. What's missing in the genotype below it? There's no asterisk. There's no way to get a green worm if you start dealing with that second worm, this one right here. You're never going to get a green worm if you start working with a worm that doesn't have the GFP gene in the first place. So what are we going to do to this worm? What sort of a cross would we want with that worm? You can self it, or you could cross it to its sibling. You could cross it to one parent or the other parent. Those are all the worms you have. You started with 2 parents. You made some kids. So now you've got 4 different types of genotypes of worms on hand.
Wouldn't you want to self-cross it because it has an asterisk and a negative, and none of the other ones have both of those together?
That makes sense to me.
Would it be better would it work to self-process to parent 1 because you can get 2 parents that are full homozygous rollers, and then those would just be, then you can have the heterozygous for the asterisk still, so you have the roller completed?
Okay, so this is the difficulty in answering this question, which is not a question type I would normally ask on an exam, is because of this word efficient. That's what we're discussing right now. That's what geneticists that are actually working on breeding programs want to do. We want to make this target genotype the fastest we can, get it on market, get it out to people that want it. So now we have to figure out what sorts of crosses do we want to set up. Either way could work. So both of you are right. You could do it that way. The question is which one's faster. It sounded like with your approach, we would need at least 2 generations. So the question then is the first approach faster? Do we get, with just selfing this individual, if we self that individual, do we arrive at that genotype in a single generation? So I'll leave that to you to work out. And basically what that's going to require is looking at the dihybrid Punnett square. If you're selfing a dihybrid, that top genotype. So I'm going to open a Socrative quiz right now. It's just a simple single question. It's quick. Don't put too much thought into it. And then I'll see you next class. All right. There you go.